Hi, my name is Zarin Azami. I'm a scientist at ONI, and I'd like to thank you for joining me in this snippet on how we can help bring a novel dimension to your CAR-T characterization. This figure is of itself a CAR-T cell, kindly provided by Dr. Nathan Singh at the Washington University School of Medicine and imaged here at ONI headquarters. It nicely illustrates the extraordinary information on CAR molecule number and nanoscale distribution that is completely lost from conventional light microscopy due to the principles surrounding the diffraction limit of light. Super-resolution microscopy refers to the constellation of imaging modalities that have allowed us to overcome these constraints really only recently in the last decade. And in this brief talk, I'll show you how ONI has helped us strip away all of the pain points and the fussy fuss of super-resolution microscopy and microscopy in general with our instruments, our sample workflows, and our analysis tools. Now, here's a closer look at the stars of the CAR-T Summit, the CARs themselves, shown at the single molecule level. As you can imagine, the workflow that we have implemented here extends to other biomarkers that interest you, particularly those that occur at low copy number, since these would otherwise be undetectable with conventional light modalities such as flow cytometry. So how do you get such a super resolution image? Well, I'm showing you here a screen grab of an acquisition that I took. Basically, here you're looking at the diffraction limited image of a fluorescently labeled cell labeled for cause. The secret to this particular modality of super resolution imaging called D-STORM, or direct stochastic optical reconstruction microscopy, works on the principle that in the right buffer condition, with a bit of laser power zipped into there, you can get the molecules, each of the fluorescent molecules, to report back their position by appearing temporally separated in time as blinking events. And then our localization algorithm can find the center point of each of these spots and mark that computationally to build up the image over time rather than get a blank sheet of light back from all the molecules that are crowded in space. You get individual molecule positions denoted from this random distribution of blinking events that happen that report back the position of each molecule. And we have more of this information available on our website. You can follow some of the webinars. In fact, I even have a webinar on there. But what this whole video sequence showed you was that this kind of acquisition can take place, for instance, to get to this picture in about 5 minutes. And we have many workflows that work on throughput and so forth. But the workflow that I'd like to show you next is then how you can take the raw data from here and put that into our analysis pipeline. And I also am very excited about showing you this pipeline because it is a collaborative tool that's available on a web-based server. So here it is. Cody is our analysis software. We're able to upload our data sets either manually, or you can actually use an uploader that we have, and you get all your localization data in here. You can adjust, you can do filtering, you can adjust for drift correction, you can identify a region of interest you want to focus on. And here I'm running what we call our clustering algorithm. This clustering algorithm finds where the localizations are and how they relate to perhaps individual molecules. But furthermore, by adjusting the length scale here with this length scale slider, you can start looking for nanodomains, how the localizations or the correlate to molecules, which then may correlate to higher order nanodomains, shown here in multicolor. And here I'm flipping on and off the filters of the clusters over the localizations themselves. Furthermore, you can run counting tools where you can find a sense of how many molecules or localizations or whatever features that you're looking for within certain regions. Here I'm showing you the counts that are coming back for that particular region there. And there's a lot we can do basically, but the great thing is that you can see everything I've run, and that list of everything I've done is collaborative. So you can see all the executions I've run, every other execution other users have run, and then you can see the gallery of data that all of the people who wanted to publicly display their particularly highlighted data sets, they can do that here. And you can of course keep data sets private. We have features called projects that allow you to keep it within your teams, but also make it really easily available so that you can share your data, share it with people who want to analyze your data. And it's really a great conduit towards things like citizen science and so forth. So it's very exciting. platform, I hope that you can just go out there and start looking at the data sets that we've already uploaded there, and our users have uploaded there as well. Thanks for listening today, and I hope this gives you a taster for what we can do with an nanometer in the CAR-T space. And we look forward to hearing from you soon. Thank you. Bye-bye.